Hello and welcome to Framing the Foundation podcast. I'm your host, Sid Parulkar. At this time, we'll be tweaking the format a little bit, not only talk about their past experience, but also touch base on some of the pressing issues and challenges that we are seeing in the industry. At the same time, we'll explore new trends that are going on in the industry. So it will be a lot much more educational for people listening into this podcast. I'm excited to welcome Dr. Konstantin Glebov. He's the Managing Director of Exosome Consulting. Recently, Park Clinical and Exosome Consulting got into a strategic partnership. Dr. Glebov brings extensive experience in exosome technology and extracellular vesicles. He has been, through his company, he's been supporting a lot of biotech ventures and investors to commercialize extracellular vesicles or exosomes. He is located in Europe and has a collaboration across Europe, has led some cross-border projects. With a PhD in neuroscience, he is very passionate about exosome technology and the exosome translational field. He is frequently a speaker, and you will see him speaking at some industry events and symposiums. He introduced the Exosome 2.0 concept at the ISCT Annual Meeting in Vancouver, where he presented the commercial framework at a global forum. So very excited to speak to him today. We'd love to talk to him about how the exosome technology is growing. So let's just dive in. Konstantin, thank you so much for joining the Framing the Foundation podcast. We have been talking about this, scheduling this for a while, but I'm glad we were able to get our schedules aligned to be able to schedule this talk. For everybody that's listening in, I just gave you a brief introduction about yourself, but would love to hear about how your journey is. Again, just a background for people watching this podcast. The Framing the Foundation podcast is more focused around people's experiences in the industry and how they've evolved. But this time we will take it a notch further and talk a little bit more about Konstantin's background and how the Exosome 2.0 version is really evolving. But Konstantin, would love to hear about your background, how the journey has been so far.
Yeah. Well, thank you, Sid, for invitation. It's a great pleasure to be here. So I am a managing director of Exosome Consulting, which is a boutique consulting company that specializes exclusively on extracellular vesicles. Majority of people know extracellular vesicles as exosomes, hence the name. I've been doing vesicles for 15 years or so, and I have seen a lot of inefficiencies in academic research, and then I have noticed that actually industry is also lagging behind other therapeutic areas like CAR-T or ADCs and so on. So I decided that maybe there is a space for improvement. Mm-hmm. And this is where Exosome Consulting came to life. And so far the journey been exciting and crazy sometimes.
Yeah, and also wanted to mention for viewers looking into this podcast is Konstantin was introduced. We got introduced through a common connection, and because of my background working at Kodiak Biosciences, which was also an exosome focused company, we had a lot to talk about. A lot of discussions led to us collaborating. But again, I'm glad that we are making progress on our collaboration as well. But the exosome space has really been dormant for a while. I think there was some level of enthusiasm, but now I think it is probably coming back. I don't know, Konstantin, what your thoughts are on how the space is evolving, and then tell us a little bit about your Exosome 2.0 vision as well.
Yeah. Well, you're absolutely correct. So the exosome space is actually finally past that hype stage. So I think the hype stage for vesicles was 2015 to 2021, 22. Mm-hmm. So now in 2025, we're kind of more to the ground with realistic expectations and with realistic promises. And most of the time, like if you know other industries and like think of Apple, the company. Mm-hmm. So exosome space at the moment reminded me of Apple in the 90s. So they were reliving on their hype from the successes from the 80s. There were a lot of things. They were still unfocused, close to bankruptcy. And this is actually where I see a lot of parallels with the vesicle space. So I really hope there will be a Steve Jobs of exosomes who will reboot the space. And luckily, I see a lot of signals that this is going to happen. Maybe not 2025, but in the next 2 years. And if this will not happen, then we should be a little bit concerned. So if we talk about Exosome 2.0 and we think that 2015 to 2021, 22 was the Exosome 1.0. This is where there was a heavy focus on so-called naive stem cell vesicles. There was a lot of promises and like miracle cures and all that stuff. And the problem was that there was a regulatory ambiguity. No one actually knew. Though we know exosomes in with different names and different forms and shapes from the early 80s, like the real research into vesicles actually happened over the past 15, 20 years or so. So there was this regulatory ambiguity as well, and that actually led to proliferation of a lot of off-label products or cosmetic products. Right. And those spaces, they somehow eroded credibility of the vesicles with regulators. There were some data gaps. There were inconsistent characterizations. People didn't know what they were doing. And it was actually during COVID that I gave a talk and I compared vesicles to the willow bark brew. But before we got aspirin, people actually collected bark of a willow tree and then brew to get the same salicylic acid. So that was actually reminded me with vesicles that we kind of cooked those concoctions. They did work somehow, but we didn't really understand how it worked. So now we're moving from that willow bark extract to a real API with a real mechanism of action where we're starting to understand. And this is what I proposed as the Exosome 2.0 last year at the ISCT meeting, that we have precise engineered vesicles. We have tissue targeting. We have defined mechanism of actions. And this is actually what everyone would love, because regulators would love, investors would love, and obviously big pharma would love that clarity, transparency, and of course, related connected efficacy that would come out of this. So this is where the Exosome 2.0 came to life. And after that, I've been working actually quite hard with big pharma and with startups and with everyone else actually to bring this to life.
Concept to life, yeah.
Yeah. So what can we do with this? Because, I think there is still a lot of potential and there is an unexplored, untapped potential, not the hype stuff, but the real efficacy. So I think we're going to solve this in the next 2-3 years, maybe, if investors would be patient enough, of course, because that's boils down to investors and competition. It's hard to compete with CAR-T that's been a success, ADCs, antibodies, and antisense therapies, and all that stuff. So either we start delivering efficacy, not only promises and science papers, but hard efficacy data. And if we will be managing this, then of course vesicles have a fantastic future.
Constantine, just want to pick your brain and probably just to cut ball on the nanoparticle technology and vesicle. Are they very similar in terms of how it, not only mechanism of action or releasing the drug, but it's more about also scaling up? Any thoughts there?
Well, there are actually more angles to vesicles than to LNPs, for example. We can always see LNPs as delivery vehicles for some pharmaceutical therapeutic cargo. But in terms of vesicles, actually, we have more sides to that than just a delivery vehicle. So we can obviously use them as delivery vehicles because we can engineer them, we can load them after manufacturing them from cells. But at the same time, if we're using engineered vesicles from, I don't know, stem cells or from other cell sources, they actually carry intrinsic functional cargo. And this actually what could provide additional benefits. So actually, I see a lot of differentiating factors from LNPs. So one can say that they can be seen as LNP 2.0, but I think they're way more than that. They're better than that. They're safer than that. They allow a lot of flexibility. And of course, if you will crack how to generate a cell that would produce your biologics and will package them into vesicles, you will have a fantastic CMC and manufacturing as well. Because you have only to engineer cell will be more or less stable, and it will produce constant flow of those vesicles. And with LNP, yes, you can manufacture them, you can precisely engineer them, but they have all the drawbacks. And we have seen this over the years that toxicity, for example, is an issue. And this we should not have with vesicles. So yeah, I see they as the next generation of delivery
mechanisms,
vehicles. Yeah. But I believe they are way more than that.
Okay, that's interesting. Just love to get your thoughts. So 5B2, I think a lot of formulation changes to an existing drug that is losing its patent. I think generics are also moving in that direction. Any thoughts there on the synergies between vesicles and how it could be implemented in 505(b)(2) kind of applications.
Sorry, the connection was not so good. I couldn't
Sorry,
hear.
in terms of like 505(b)(2) regulatory pathway. So repurposing drugs, do you see a potential there for vesicles as well? Because I was working for a company probably 8-9 years ago, and dexamethasone was being encapsulated in a vesicle
Yeah.
and delivered. So that's where the question is coming from. Like, do
Yeah.
you see synergies there? Or
It's a very good
potential?
question.
Yeah.
It's a very good question. So the thing is, I would love to see some studies, really controlled, placebo, randomized, blind or double blind, that actually would show that vesicle plus a steroid is better than a steroid alone. Because vesicle would add an additional cost, and still not sure whether actually the efficacy would be better than the steroid alone. So I did not see this data, and I actually had this discussion with big pharma people just 2 weeks ago. It is extremely interesting. Can we repurpose something that is very cheap? Okay, not in US, but worldwide, steroids are very cheap. So they are very efficacious. Would we make them better? I am still skeptical on this. I believe that vesicles and oncology, there is a lot of potential because you can basically wrap all those chemotherapies into vesicles, hopefully improve tropism, so basically tissue targeting. Plus, you can remove or reduce toxicity. There I see kind of a more or less clear path to success and clear-cut benefits. In terms of steroids, I'm not so sure because vesicles on their own, they have anti-inflammatory properties. Everyone knows that. Can vesicles work better than steroids, or vesicles plus steroids work better than vesicles alone or steroids alone? I don't know, and I didn't see any convincing data so far.
Okay. But you see a potential there for any kind of repurposing of drugs within vesicles. That could be a new formulation that could be developed for an already patent expired drug.
Yes. But again, for the space of oncology.
Right.
In oncology, I see there is a potential. In CNS, so in neurology, there could be also potential. But in terms of inflammatory diseases and vesicles plus steroids, I'm a little bit skeptical because, again, maybe even scientifically it will be good, but then the question is commercial. Would actually the benefit justify the additional cost of vesicles? So we should not forget, and this is actually what a lot of people in academia forget, that despite good science, your discovery should actually be able to make money. And if it's not going to make money, then it should probably stay as a science.
Yeah. That's a great point. Yeah, I think that is so well said, because we have seen this over and over that great science moves into clinic, but scalability is always an issue, right? And it boils down to the production cost. How much it's going to cost to really make this. We have seen this in a few companies that I've worked in the past, and we have spoken about it offline as well. But you see a huge opportunity here, right, in terms of vesicles.
Yes.
And from a timing perspective, do you think now is a good time for companies to start thinking and moving in this direction?
I believe so. The climate, though, is a little bit hostile at the moment, so it's very difficult to raise money. Our clients manage somehow, but I know a lot of companies who actually did not meet their fundraising goals and were unable to fundraise at all sometimes. So it's very difficult. But again, it's because we still have those pains from the Exosome 1.0. So from all the over promises and under deliveries in the past. But investors actually should, okay, man, there were decisions made, maybe people lost money, maybe something did not work out. But we learned so much, even from those mistakes and those failures, that now is actually a perfect chance to enter again and to start working focused, precise, with determination and with a long-term vision. But again, on the projects that make not only scientific sense, but also commercial sense.
Yeah, absolutely. Yeah, that's well said. Konstantin, just want to understand, like, what are some of the previous failures that you've seen with Exosome that you feel should be corrected right now?
First, we have to really, we're not going to name names, of course, but if you look at the space before, heavily focused on those unmodified naive vesicles. So, and they do work. sometimes, but it's very difficult to manufacture them consistently, because in manufacturing, when we do quality control, we mostly focus on size distributions and zeta potentials and maybe some kind of key target therapeutic molecules composition there and so on and so on. But that is not enough. So what we really need to have is validated efficacy and potency assays that actually are integrated into the manufacturing. And so far, actually, I did not see that very highly implemented or deeply implemented by many companies. So we just produce, it kind of works, and then it goes to clinical trial, either FDA approved or the IRB approved. And then it actually doesn't work as good as it should be. And the question is why? The reason is because we didn't know the API. We didn't know the exact mechanism of action. And when we did not know that, we cannot control the CMC and manufacturing. So that is where the issue was, because we did not know enough about them. And this is where the failure came. And then investors, of course, lost a lot of money. They got upset and disillusioned with vesicles, and they went to other places where actually you have more clear-cut returns, like CAR-Ts, ADCs, and all the other things. So we need to move forward and focus on explaining to every stakeholder, either regulator or investor, actually what your vesicle is doing, how is it doing, and how I can ensure that during my manufacturing, scaled up manufacturing, I can manufacture vesicles with exactly the same efficacy and exactly the same potency, and obviously the same API present all the time. So this is where we failed before, because we used a lot of MSCs from various sources, and of course we all know that people all different, so there is a genetic variability, so sources will affect your final product. And even if you are kind of sure that you have a good solid donor for your cells, there is this clonal competition when you grow cells. So particular subcellular populations can outcompete and actually shift your composition slightly, but that might actually have a tremendous effect on your potency and efficacy. So this is what I see as the mistake. From a scientific point or from a startup point. There were also actually mistakes from investors' point. And I have presented this data in Vancouver last year, that actually when I looked at vesicle companies and compared them to biotech startups in general, it looks like that actually vesicles company were heavily underfunded, by a factor of 5. Okay. So there is, actually, we might say, okay, science was not there exactly how we needed it to be. But at the same time, those companies were underfunded. They did not have enough money. I'm not talking about those superstars that are few in the space, but we're talking about kind of a broader area.
Is it because of lack of confidence in the technology?
It is probably because of lack of confidence and also lack of understanding. So this is another area where actually I'm heavily involved, is educating everyone. So, like investors, big pharma, explaining why we failed, what went wrong, and how actually vesicles are great again. So the education is an important component. So they probably got skeptical because first they didn't see immediate results, because that is another data point that actually we found out, is that on average it takes, I think, well, now it's changing, but before, in the first phase of vesicles, bench to trial transition took 9.6 years or so, while in average on biotech it takes 3 to 5.
3 to 5, yeah.
Yeah. So, and this is of course is money drain, so you lose your money, you still cannot show anything.
Any reason why it's taking longer?
It took longer. I hope now it's not going to take so long, because everyone got a little bit more understanding. Regulators actually got clear understanding of what, and this will change. So this is why 2.0 is so important, that we will have a renaissance.
That's great. So, but do you see a focus or interest more from large pharma, or do you also see a potential for small biotechs to also take this on?
Well, you cannot, as a startup, as a vesicle startup, you cannot succeed without big pharma, because at the end, big pharma is your ultimate buyer, who will buy your program, who will license it, and will help you to bring it to the market, because they are the ones who got not only required capital, but expertise to bring those drugs to the market. So you cannot survive. For big pharma, it's a little bit difficult because there's so much noise and they're still interested. So I still get inquiries. We still actually work. We have projects with big pharma, but they are cautious now. So they're very cautious. So I know that before we were discussing those kind of, checks like in a million range upfront. So now we talk about hundreds of thousands upfront. So it's a big decline. So they're more cautious and they want to see data. They don't want to see your papers. They don't want to see your decks. They want to see reproducibility, efficacy, and safety. And in majority of cases, actually safety is there. So I'm not aware of any clinical trial where vesicle-based therapeutic actually failed phase 1 because of safety, inherent safety. So there might be some issues with manufacturing and then safety, but the vesicle itself, I'm not so aware and never heard of this. So I think the key issue is efficacy and reproducibility.
So do you see like a specific therapy area that could be focused more? Because I think vesicles moved from focus on oncology to getting into the dermatology space. But is there a specific area that you feel has higher potential of really adding value using vesicles?
Yeah, absolutely. So first is definitely regenerative dermatology. So this is area where we are also heavily involved. This is actually where you will see faster returns and fantastic results. So we just recently finished actually a trial on 20 patients, 20 subjects, not patients, in the regenerative dermatology space. We got hard numbers from those people and the results are very promising. So you can definitely get fantastic results and you can get fantastic returns as well, as we all know about that space and key players in that space. Another area I would love to see progress is CNS, so neurology. I believe vesicles can do a lot of good there. We still have some issues with BBB and delivery, but I hope we will solve this very soon. I have seen some encouraging data in that area. And another area that I would love to have is actually respiratory because of the mode of administration. So if you will be able to solve this efficacy and your vesicles will be stable when you will do the inhalation to administer them to the lungs, that's a fantastic space to be in, because there's a lot of interest. And potentially GI as well.
Okay.
And heart, cardiology as well. So there's some interesting developments in cardiology.
Yeah, I think when I was working in this space a couple of years ago, I think it was all oncology focused, and eventually, I think dermatology kind of took the lead in being the area where vesicles could be really add value. But yes, I think there's a potential. Again, for the audience, I think Smart Clinical and Exosome Consulting, we collaborated so that Exosome Consulting can provide guidance in terms of early clinical development, and then we can come in to help take these assets into clinic and drive the clinical studies. So it's been a great collaboration. Constantly, we continue to evolve every day. So this has been a great conversation. Anything else that you want to add about what your experience has been so far? Changing landscape.
Well, it's hard work and sometimes very ungrateful one.
Yeah.
But the space is changing and the space is moving. So, and everyone should actually understand that in 2, 3, 5 years, the vesicle space will be changed drastically. And if you do not adapt, if you do not innovate and do not progress and do not fail, that is actually what is another important thing in the space. Like you see a lot of companies and they have their pipelines published and then their pipelines get updated, but nothing progresses and fails. I raise for myself questions. Why? And I think we should fail and we should not penalize this because we learn a lot. And if you progress and fail, you stand up and you do something new and something again, then you will succeed. So this is what we don't have in this space, unfortunately. So let's progress, let's innovate, let's fail, and let's do it again. This is how we can bring the space and regain the credibility, regain confidence from investors, and again, invoke interest from big pharma because they are the ultimate buyers for all those products. So this is very, very interesting and very difficult, but it's very, very rewarding, I can tell you. Our collaboration with you, I think it would be very fantastic opportunity for everyone, not only startups, but actually for big pharma and investors as well. Because the problem is majority of those startups on vesicles, they actually come from academia and universities.
That's correct. Yeah.
Majority of them, especially in Europe, they heavily rely on governmental grants. And if your startup is relying on grants, well, is it really a startup? Why you unable to raise some money?
Yeah.
So, and this is where we can actually together help them a lot. So we can help them to build the story, because actually this is what drives this: a good story with solid data that
That's
underpins that story. So we work on a very early discovery and preclinical up to IND and then maybe phase 1. And you have fantastic expertise and projects on the clinical development. So we basically merge and marry those two expertises together, and you will get a one-stop shop for this. This is another interesting thing, because so far what I see, we have this academic product, then we have a little bit of preclinical development, then we have again separated phase 1, 2 development, and that's it. But with this partnership, I hope we will be able to offer something unique.
Right.
With a lot of insights, and as a one-stop, I think it would be fantastic, because you will get the same people to work with you on a journey.
Absolutely. I'm looking forward to, we have a few things that are in discussion, so which is great.
Yeah.
And I look forward to building this relationship.
Yeah, absolutely.
Again, on a closing note, I want to thank you for your time, Konstantin, for being on this podcast.
It's been a pleasure.
I really hope people enjoy. If I will put a link in the description when this podcast is posted on YouTube, on where you can reach out to Konstantin as well on LinkedIn. So do not hesitate to reach out to Konstantin or myself through LinkedIn, and we would be happy to help you with your projects. Great. Again, thank you so much, everyone, for joining. Thanks, Konstantin, for your time as well today.
Thank you.
Thank you.